Okay, there's only one little thing left in this chapter. I kind of went into it a fair amount with you guys on last class, but I'm going to review it again because I didn't get into it in all of my classes. So we were talking a lot about the electron transport chain. I just want to highlight this one more time because this feeds into the last little section. And then we'll have some time for questions and to go over some of the key points of both photosynthesis and respiration. So NADH, FADH2, those two molecules that were created when they picked up electrons that were stripped off of glucose, will drop off their electrons to the electron transport chain, and as they do, they get converted to NAD plus and to FAD as well. So you kind of regenerate NAD plus and FAD at that point. Where does that NAD plus go to? Like, where is NAD plus involved elsewhere in the process? Any ideas on that? So glucose converted to pyruvate, and we have net to ADP converted to net to ATP, and we have 2 NAD plus converted to 2 NADH. So right here is one of the locations. You'll also see it in the transition state. You'll also see it in the Krebs cycle. Everywhere that you produce NADH, you needed NAD plus to start with. So that's what happens there. This is where NADH goes to. It's where NAD plus originates to go back here. So NAD plus and NADH, they're shuttling electrons to the electron transport chain, and they're going back to pick up some more from more food. So that's one of the kind of concepts that I just wanted to highlight before we go forward. The other thing is, where do those electrons end up? Those electrons, after they've gone through the electron transport chain and caused hydrogen to be pumped into the inner membrane compartment, they end up binding with oxygen to form water. So oxygen is the final acceptor of electrons in this process. So without oxygen present, there'd be nowhere for these electrons to go. The electrons would back up, basically. There'd be no way to do these series of redox reactions, and ultimately NADH and FADH2 would not be able to drop off their electrons at the electron transport chain, so they would remain NADH and FADH2. So this plays into this next place. So Yep. They just feed into different locations. So here NADH feeds in through 3 pumps, and FADH2 only feeds those electrons through 2 pumps. And that's why NADH makes a little bit more ATP than than FADH2. But yeah, both of them, they ultimately both end up at oxygen. Okay, so without oxygen, you'd have nowhere for those electrons to go. So one, you'd be producing no ATP through this process because you wouldn't be continuously building up that concentration gradient. And 2, NADH and FADH2 would not have anywhere to drop off their electrons. Okay, so that's going to set up the next thing. So there are ways of generating ATP that do not require oxygen. So, and these are called anaerobic respiration pathways. And we're going to talk about one on this page and then 2 kind of on the next slide. So one way of doing anaerobic respiration is to use an electron acceptor that is different than oxygen. So you can use something else that will fit at the end of that electron transport chain to take those electrons off the electron transport chain that will not be oxygen. And some organisms use things like nitrate, sulfate, and CO2. You don't need to remember those specifically at all. But there are other possibilities besides oxygen to pull electrons from the electron transport chain. However, oxygen is a strong oxidizer, which is why we call it oxidation. And oxidizers are really good at pulling electrons from other molecules, So oxygen is pretty much the best at doing that. It's not very large, it's got a fair number of protons, so it's pretty darn electronegative. And it's going to be pulling electrons really fast. Whereas nitrate, sulfate, and carbon dioxide, though they'll pull electrons, they're not quite as strong at it. So the electron transport chain will run a little bit slower with them. You're going to generate less ATP, or ATP at a slower rate, using one of these other electron acceptors. For some organisms, that's fine. They generate plenty of it, and they can now live in an environment that does not have oxygen, that has something else, so live in an anaerobic environment, and that's fine for them. Other organisms actually have an option of either respiring with oxygen or respiring without an electron acceptor at the end there. And these organisms, they're often called facultative anaerobes. What they can switch between the two mechanisms. Yeast is an example of these. We are actually an example of these as well. So they can do an alternate pathway that does not actually use the electron transport chain. They are going to take the electrons from NADH and convert them back, and to convert NADH back to NAD+ without actually generating any more ATP. And this is a pathway called fermentation, and we're going to talk about two different mechanisms of fermentation. So anaerobic respiration can have an acceptor besides oxygen to run the electron transport chain, or it can use the electrons from NADH to actually convert pyruvate into something else. And in that process, it's basically getting rid of NADH, and it's getting rid of pyruvate without generating any more ATP. So it's wasting a lot of energetic resources. Pyruvate has a lot of energy, NADH has a lot of energy, but it'll allow it to continue doing this first step, glycolysis, in the absence of an electron acceptor like oxygen, nitrate, sulfate, or carbon dioxide. So fermentation, or fermenters, so organisms that do fermentation, will acquire their ATP only from glycolysis. So glycolysis, which is this process, you have glucose being converted to pyruvate, and in the process you make a little bit of ATP. This requires glucose to be present, ADP to be present, and NAD+. And remember, how we generated NAD+ was the electron transport chain. If the electron transport chain is not functioning, you need another way to convert NADH back to NAD+ to allow glycolysis to continue. And here are two mechanisms for doing that. So fermentation is an alternate reaction to regenerate NAD+, which can then be reused by glycolysis to net 2 more ATPs. So fermentation, the only thing it's doing that's productive in terms of energy is it's regenerating NAD+. So it's an alternate mechanism to the electron transport chain for doing this. So here's alcoholic fermentation on the left here. And alcoholic fermentation takes your pyruvates, uses the energy from the electrons of NADH to convert pyruvate into ethanol, and that regenerates NAD+. It also loses carbon dioxide at the time as a waste product. So this is something that we exploit as humans. We exploit the waste products of this. The waste products of alcoholic fermentation are carbon dioxide and ethanol. The real reason for doing alcoholic fermentation by yeast is the NAD+. They don't care about carbon dioxide or ethanol. So carbon dioxide we use in baking to make breads rise. So we feed sugars to yeast, and they give off carbon dioxide as a byproduct of respiration, and that causes the bread to rise. If we're making alcoholic beverages, beer, ethanol, beer and wine and anything else, we're exploiting the ethanol production of yeast. So we feed sugars to yeast, either sugars in terms of grape juice or sugars in terms of sugars that have come off of barley and other grains for beer. And the yeast go about producing ethanol. So in alcoholic fermentation, why do you think brewers take their big vats of wort, so that's the sugar water that they've made up, and they throw in the yeast, and then they put a lid on the top and put an airlock on it? What do you think they're trying to prevent from happening with the airlock? Well,
the oxygen getting in.
Oxygen, yeah. So they're preventing oxygen from coming in, ultimately to prevent oxygen from being an electron acceptor and driving aerobic respiration. Why would aerobic respiration be bad for a brewer? Okay, yeah. So aerobic respiration produces water as an end product, whereas fermentation is going to produce ethanol, and they want ethanol, right? So yeah, they keep oxygen out. That's going to allow ethanol to be produced. So that's what yeasts do. That's one of the mechanisms. So they can switch back and forth, which is why if you add lots of oxygen, then it can stop this process. That's actually something that brewers do at first. They add lots of oxygen, causes the yeast to grow really fast because they're producing a lot of ATP, but they're not producing any ethanol. And then they seal out the oxygen and cause them to switch over to fermentation once there's been a lot of cells made. And then they convert all that sugar into ethanol. Ethanol is not the goal of yeast. You ever wonder why wine tends to be around 12% alcohol content? Wonder, what do you think 12% alcohol does to yeast? And why they would stop fermenting at that point? It's deadly to them. Kills them. So this is why wines tend to be sweeter than beer. Beer, it's only like 3% because there's not much sugar in there to start with. Ferments it all out. The yeast is still alive, and then home brewers will put more sugar in to make carbon dioxide to make it bubbly, whereas wine, they ferment until the yeast dies. So ethanol is toxic, so it's not something they want to make. They're making NAD+. So do we do fermentation? I said we do. Under what conditions do you think we would do fermentation? Anyone know when lactic acid is produced? All right, it can be exercise. So you've heard of aerobic exercise. There is such a thing as anaerobic exercise, basically. What happens, so let's say you go for a jog. So you're going out, you start running, your muscles are demanding more ATP. So what happens in your body to allow your muscles to produce more ATP? So the demand of, so you're producing this much ATP, your muscles now need this much ATP. So what happens to your heart rate and your breathing rate? They go up. And why are they going up, do you think? All right, they're going to supply your muscles with more oxygen to increase the rate of aerobic respiration to match your muscle demand. And say you run a little bit faster. Your breathing rate and heart rate go up a little bit more. So then you reach a point where your heart is pumping as fast as it can, so you reach your maximum heart rate, and your breathing rate is as fast as it can. So you're getting as much oxygen as you can possibly supply to your muscles. And let's say now you're being chased by something. So you need a little bit extra boost. So your options really at that point are stay at that pace or slow down and possibly be caught by whatever is chasing you, or somehow make up for that difference. So you're producing this much ATP through aerobic respiration, but you need to produce this much to be able to go faster. So what you can do is you can take some of that pyruvate and convert it to lactic acid to regenerate NAD+, and feed that NAD+ back into glycolysis to produce a little bit more ATP. So you're still going to be doing aerobic respiration at this pace, but the excess NADH, which has nowhere to go because you can't supply any more oxygen, you'll burn up so you can do glycolysis and just give yourself a little bit more ATP to get going. So we would never survive on this alone, which is why you hold your breath for too long, you're going to pass out. But you can use this as a way to supplement the amount of ATP you're producing through aerobic respiration. So have you ever heard of blood doping? Anyone know what that is? All right, what is it? All right, not exactly, but kind of. One is athletes will take blood out, actually, and regenerate the red blood cells, and then before an event, they'll put the blood that they took out back in. So they're not directly injecting oxygen. What they're doing is they're giving themselves more red blood cells, which makes their blood able to carry more oxygen, so that every time their heart pumps, they're moving more oxygen through their body. So how does that help an athlete? Okay, they can stay in aerobic respiration for longer. And remember, aerobic respiration produces 34 ATP per glucose molecule. Fermentation and glycolysis produces 2. So much more energy produced through aerobic respiration. So athletes also can do this by training at altitudes. So that increases your red blood cell count. Sleeping in hyperbaric chambers is another thing athletes have tried to do, and that's simulating high altitude. So it causes their red blood cells to increase. Very hard thing to detect because it's your own blood that you're putting back into you, right? But there have been a lot of athletes that have done things like that, right? This is also why football players, you'll see on the sidelines between plays, sucking on oxygen masks. They're trying to get themselves back into aerobic respiration. They're trying to also get rid of some of the lactic acid because that can be damaging to your muscles if there's a lot of it in there. So they'll push themselves into the anaerobic, and then they'll try to bring themselves back into aerobic, producing enough ATP to keep their muscles going. So when you do aerobic exercise, you're often looking at your heart rate. You're trying to keep your heart rate below the maximum rate so that you know you're always in aerobic respiration. Because for the long term, that's going to be better health-wise, because it's using more processes. It's going to burn more fuel. This process, glycolysis, really only burns very easy to get fuel, so simple sugars, simple carbohydrates, whereas if you remember from that graphic I showed with all the different pathways, a lot of like fats and proteins will feed into other stages of cell respiration that do require oxygen to get into. So if you want to burn fat, you want to do aerobic, not anaerobic. Okay, so what's the whole goal of fermentation? NAD plus, right. So NAD plus, this molecule here, that is the whole goal of fermentation. So fermentation regenerates NAD plus. It's an alternate mechanism for regenerating NAD plus, allow glycolysis to continue in the absence of oxygen. Fermentation alone does not generate any additional ATP. Where is the ATP produced in fermenters? Just in glycolysis, yeah. So that's kind of the take-home message there. Okay, so let's see if you can answer this one. Okay, what is an advantage of anaerobic respiration over aerobic respiration? All right. Okay, most of you said A, and you'd be right. So anaerobic respiration, which, remember, could be glycolysis coupled with fermentation. That would be a form of anaerobic respiration. Or it could be glycolysis followed by all the rest of the cycles of aerobic respiration, but not having oxygen as a final electron acceptor. Both of them allow organisms to generate ATP, even if it's just a small amount, when oxygen is not present. Anaerobic respiration definitely does not generate more ATP per glucose than aerobic cellular respiration. In most cases, aerobic is higher. Aerobic respiration will also produce it at a faster rate, even if you're using an alternate electron acceptor. Both of them produce CO2, except for in humans, lactic acid fermentation doesn't produce CO2. But CO2 is not really toxic in the quantities that you'll produce it at. It just diffuses right out. Anaerobic respiration gets rid of pyruvate. That's true, but that's not really the reason why it's an advantage. Pyruvate itself is not going to be too harmful. The real reason is that it generates ATP even without oxygen. Okay, here's one more question, and then we'll get into some review just to see. This one's kind of a longer one. So let's say you're growing some cells in culture. Cells grow really well for several weeks, and then they don't seem to grow as well. You conduct some tests and determine that there is a lot of lactic acid in the culture fluid. Which of the following is the most likely explanation for why the cells are not growing so well? Looks pretty good. Okay. So most of you said D, and you'd be right. So there is not enough oxygen. So why would too little oxygen lead to lactic acid production? All right. Lactic acid production is a product of lactic acid fermentation. Lactic acid fermentation occurs when there's not enough oxygen to run the electron transport chain, so it gets rid of that NAD, convert it back to NAD+ to allow glycolysis to continue. If there was not enough glucose in the culture fluid, you wouldn't get any lactic acid production. You would just get cell death for the most part. If oxygen was plentiful, you're still going to do aerobic respiration, so you wouldn't have lactic acid. So lactic acid, or in the case of yeast, ethanol, is a sure sign that there's limited oxygen resources. So D. Okay. All right, so that's the end of this chapter.